Good afternoon. I hope you're doing really well today. I thought I'd share with you this donut mitosis. You like donuts? Wouldn't that be a nice thing, hey? Anyway, before we talk about mitosis, just another reminder that we do have the midterm coming up.
I think you might have accidentally muted yourself.
Oh, there we go. Thanks for letting me know. Yeah, so it's going to be similar to the last midterm in terms of the format. It's on Moodle. It's using the lockdown browser, and it's just different topics. So this time it's topics 7 to 12. So 7 is the cytoskeleton, and 12 is the one we're covering right now on the cell cycle. So a few less topics, but it's a pretty information dense kind of topics. So make sure you get started on that. There are on Moodle a number of resources for you now. I have a sample midterm. I have some sample questions up on Moodle too, just like last time. I think I found 70 questions just from a test bank. They're not going to be easy, these questions. You won't be able to answer them probably without studying, at least a little bit. And I have the Kahoots I've also put up on there. And so you can access those if you want to try them out again. So Monday is going to be a full review session. And so hopefully by then, though, everyone's at least taken a little bit of a look at their information, and Monday we'll be able to help polish things up for you. If people have anything they want to cover in particular on Monday, please let me know ahead of time and I can include that into the review session. All right, so let's talk about mitosis. Sometimes I ask people about the stages of mitosis, and most people know this one. Hold on, there we go. Prophase, Metaphase, Anaphase, and Telophase. And my high school teacher told me this little mnemonic here: play me a tune, and that way you can remember what order they are in. Most textbooks, though, are starting to include pro-metaphase in there, and sometimes they call it late prophase, and because they're really just trying to distinguish that there are a number of things going on, and some things happen kind of early in prophase, some things happen in late prophase or pro-metaphase. So our textbook is going with that model, so that's what we're going to go with as well. So I'm hoping that the stages of mitosis are reviewed for you. It's usually not too complex, and like I said, it gets a little bit more complex when we get to the regulation and when we start talking about meiosis, and some of those things we're going to cover today in this class. So here's interphase, right? So remember what happens in interphase. It's going to grow. So it has G1, which is growth 1, and then we have the S phase, which is the DNA synthesis, and then we have G2, which is more growth, and so this is late interphase. And so one thing to note is that in late interphase, the centrosomes here, they do duplicate. So these are microtubule kind of seeding origin sites, and the DNA is still kind of diffuse and not condensed. So take a look at this micrograph on the right. I just want to point out that the DNA is stained blue. That's the DNA. And the microtubules here are these green things. So microtubules, or you can call them spindle fibers. Those words, I expect you to be able to use those words interchangeably. Intermediate filaments are there kind of for structural support. We're not really going to pay too much attention to them in these diagrams here. So prophase, what is happening in prophase? So two main things are happening here. Notice the chromatin has now condensed, and so now we have something that we call heterochromatin, meaning it's condensed, or sometimes people use the word and they just say it's condensed into a chromosome. It's kind of funny language because chromosome doesn't necessarily mean condensed, but often it does when we're talking about mitosis. So I just like to say it's condensed chromosome rather than just saying it's a chromosome. So notice the spindle fibers are starting to form. So the two centromeres are centrosomes, sorry, are the two centrosomes are separating from one another. And so as these stretch out, they're actually pushing each other. As they stretch out, they're actually pushing each other apart. And there's also a little bit of pulling because these centrosomes are actually anchored to the membrane. So a little bit of pushing, a little bit of pulling, and during that is going to allow the cell to begin to elongate a little bit. So notice there's still a nuclear membrane, and that's kind of the part that happens in late prophase or prometaphase. The nuclear membrane is eventually going to dissolve. So there's prometaphase, and the nuclear membrane is dissolving. Remember, we were talking about these nuclear lamina. The nuclear lamina are intermediate filaments that are holding the membrane together, kind of like a scaffold. Well, they just get disassembled. And so the nuclear membrane is a really large membrane. It's not really that structurally sound without those intermediate filaments. So it kind of blobs apart, and you can see this is kind of in this diagram, they look like little blobs. And as a consequence, the spindle fibers can now invade that nuclear space and start to attach to the centromeres. Remember, the centromere is that attachment point kind of in the center of those, it's in the midpoint of the sister chromatids. So they're attaching, and they're starting to move around is what's happening here. So metaphase, a big M for metaphase, and M is also for middle. And so all of the chromosomes eventually get lined up along the middle, which also has a name. It's called the metaphasic plate. So that's metaphase, right? And it's may not look like much, but this is actually a very involved process, and there's all sorts of checks and regulations here, because what we want to do is it's very important that the cell makes sure that every single spindle fiber is attached to a chromosome. Every chromosome is attached in the proper places, and everything has to be lined up and accounted for. So it's very important before you actually do the separation that it's done right, or else we're going to get really badly mutated cells. We have to have the right chromosomes all lined up. So there's a lot of checks and balances kind of in this thing, and it talks about them a little bit in the textbook. In the future, if you take Biology 201 at University of Alberta, you're going to talk a lot more about the regulation process in terms of what is going on here and all the checks and balances. So metaphase, middle, everything is lined up. So anaphase, A is for action. So now that everything is lined up properly, there are little proteins that are found at the centromeres that are holding those sister chromatids together, and they get dissolved. And the spindle fibers are going to tug those sister chromatids to each of the poles of the cell. And so this is done right, you're going to end up with one set of DNA to one daughter cell and one set of DNA to the other daughter cell. So telophase is kind of, okay, we've done it. Let's clean things up now. So you reform your nuclear membrane, and usually at this point, sometimes cytokinesis is starting. Actually, cytokinesis, we'll talk about that in a moment. Actually, so in most cells starts actually during anaphase, before these things are all separated. So like I said, telophase is mostly the nuclear membrane reforming. Again, there's always these exceptions in biology. It really kind of depends on the tissue and the cell type and all that. There are some cells that never really do a full telophase. There are all sorts of mushroom and fungal species that have these multi-nucleated cells, or they kind of end up with some weird things going on. But in human cells, usually we go through telophase, and that's the reforming of the nuclear membrane. So I'll show you this video. This is a video of an animal cell, and it's time-lapsed. I think this is 16 hours time-lapsed into about 50 seconds. And you can see it starts basically at prophase with the chromosomes being condensed. And most of this is actually metaphase, where it is attaching spindle fibers and basically lining them up at the cell pole. And you're going to see once everything is lined up, anaphase, it just boom, it's going to happen very quickly. So get ready for it. Almost ready, getting there. You can see that one last chromosome is having a hard time getting lined up. There we go. There's anaphase. And you can see, like I said, the cytokinesis, it's happening almost immediately, even during anaphase. So very nice. There's a few of these on YouTube. This is one of the better ones. So just a couple of reminders about the spindle fibers. So I just wanted to point out some terms here, right? So this is the centrosome, and sometimes you're seeing a lot of places call it the aster, because aster means star. So it kind of looks like a little star, and you can use either of those words interchangeably. So remember that the spindle fibers are made out of microtubules, so that's a good thing to know. And the last thing to remember is that these attach here, and they attach at the centromere. So just trying to remind you of the terminology here. The other thing to remind you is these things here, these kinetochores. These are motor proteins. And these motor proteins, just like all motor proteins, they consume ATP and they move something. In this case, they're moving chromosomes along the microtubules. So they're not really shown very clearly here in this micrograph, but they're kind of very similar to the dynin and the other motor proteins that we talked about before, where you can almost picture them as like little legs crawling along the microtubules and moving their object. So in this case, like I said, the object just happens to be a chromosome. So let's talk about cytokinesis. So cytokinesis is technically the division of the cytoplasm. And it's sometimes part of mitosis, sometimes it's not. There are some cell types that undergo all of mitosis, and then cytokinesis happens later. In human cells, most of our cells, it happens simultaneously. Cytokinesis happening, like I said, it kind of starts somewhere around anaphase and finishes by the time telophase is done. That's not true for all human cells. By the way, muscle cells are sometimes multinucleated and don't ever, some of them don't do a full cytokinesis. And like I said, there's lots of weird things that go on, particularly if you're a mushroom. So in an animal cell, we have this pinching that goes on of our membrane, and this pinching is called a cleavage furrow. So I have some images of that for you. Here's a cleavage furrow in an animal cell. And so this is done by microfilaments. So remember the microtubules, that is spindle fibers. Now we're talking about microfilaments. Remember that microfilaments are made out of actin. So actin is the same stuff that is in our muscles, and it's the same stuff that we see in amoebas, and it's a part of the cytoskeleton that is close to the membrane, so kind of just found just underneath the plasma membrane. And so this is why it's a good instrument for pinching on the membrane along. So the cleavage furrow is just that pinching of the membrane, and eventually you get two daughter cells. Another picture or two showing the top one is a sea urchin. There's a lot of cell biology done in sea urchins. I think their cells are easy to culture. The bottom one, it looks like some cancer cells. Also, cancer cells are easy to culture as well. So what about animal or plant cells? We can't do that. We can't pinch a plant cell because it's got a cell wall. So we're doing this a little bit differently. And in a plant cell, we're going to make something called a cell plate. So what we're looking at here, you can see it's labeled here. We've got these little vesicles, these little blobby things. So if you remember what a vesicle is a little membrane compartment, and it's made by the endoplasmic reticulum. And so that's exactly what we're doing here, is we're making some proteins in the endoplasmic reticulum, and they probably go through the Golgi first. And the Golgi says, where is this thing going? And this thing is going to the cell plate. So I'll show you a couple other cartoons, and then I have another image or two of a cell plate to show you. So what proteins are we talking about? Well, it turns out these proteins are enzymes that make new cell wall material. So if you remember that a plant cell wall is made out of cellulose, so these are cellulitic enzymes, meaning they reconstruct and make cellulose. So eventually they kind of merge together, and this merged together thing is called a cell plate. And they import in new carbohydrate and eventually make that cellulose layer, and then you get two cells. So a little bit of a different system because you had a cell wall, so you got to do things a little bit differently. I'll show you an image. I really like this set. This is that set we used in lab, I think it was lab 2. We're looking at the onion root tips, and you can see the cell plate is right down there in the last 2 pictures, telophase and late telophase. I also want to show you this one here because I found this on the internet, and it's almost laughable. I mean, look at the typos here. We have interphase, we have prophase, and then prophase spelled entirely different. In fact, this is prometaphase. Anyway, it's just kind of laughable, and the different font sizes and all that. It's just, every time I see it, it gives me a little chuckle. So I thought that I would share that with you. So I do have a video. I'm not going to play it for you. I just want to make sure I have time for everything else today. So you can look at it at your own time, but it's a review of mitosis. You can take a look at the PowerPoints and just click on the link for this slide and you can play the video. So what I wanted to show you today is on the exam, you may have noticed that there's going to be a mitosis meiosis question worth 5 marks. So some of you are probably wondering, okay, what's that all about? So I'm going to show you the mitosis part right now, and then I'll show you the meiosis part in a few minutes once we're done talking about meiosis. So what I'm going to do is I'm going to give you some information about a cell, and I'm going to tell you to draw some of the stages. So you don't need to draw everything. I'm just looking for the number of chromosomes and what the spindle fibers are doing. And it doesn't even have to be very nice. It just has to, you just have to be able to count properly. Most people find the mitosis part pretty easy. It's when we get to meiosis, it sometimes get a little bit more confusing. So take a look at this information here. We have a diploid cell, and we have n equals 3. So that's the haploid number, right? So that means that the haploid cell has 3 chromosomes, right? So 3 chromosomes. Okay, I'm running out of space. There we go, chromosomes. So what does that mean the diploid cell has? The diploid cell is 2n, so that's 2 times 3. So that's 6 chromosomes. So that's a half a mark out of 5 if you can just figure out how many chromosomes. Okay, it's going to be a word question, so you just got to read it and figure out what it's saying. Okay, and I'll show you another example or two when we do a review session on Monday. So, and then it's saying, okay, draw interphase, metaphase, and the daughter cells. So I'm going to do that on the next slide because I've made myself a little bit more space on that and give you an idea what this is going to look like. So remember this magic number, 6 chromosomes. Okay, so let's do that, shall we? So here's my diagram. I will, what you're going to do on the test, by the way, is normally, obviously, we have a paper test. You're doing this on Moodle. So on a blank piece of paper, you're going to do this particular question, and then you're going to email it to me and make sure you get it to me within 10 minutes of the completion of the test. And so just take a photo with your camera or scan with your scanner and send it to me, and just make sure it's clean enough. I don't want anything like super blurry. There should be no excuses there. So n equals 3. Remember, we want 6 chromosomes. Okay, so that's my magic number. So interphase, I just want to see that you can count, right? So I'm just draw 6 chromosomes. 1, 2, 3, 4, 5, 6. Remember, I'm asking you to draw chromosomes and spindle fibers, and you don't have to be fancy or artsy or anything. So remember what happens in interphase. Those are going to duplicate, and I'm just going to draw a little aside over here. They're going to replicate, and in prophase, we're going to end up basically with 6 sets of sister chromatids. So this is just me being a little more artsy. If you want to draw them like that, or if you want to draw them like this, that's fine. Just show that they're connected, those chromatids. So I need to draw 6 of those. And in the second picture, I need to draw them at metaphase. So here goes. So remember metaphase, M for middle, and I'm going to draw 6 of these. So there's 1, 2. If you want to be a little more artsy, you can try to squeeze them in like that. There's 3, 4, 5, and 6. Okay. Let's not forget those spindle fibers. Let me see if I can figure out how to change the color of my pen. And I'll draw the spindle fibers in another color. Here we go. So the spindle fibers, of course, are going to the poles. So I'm going to have a spindle fiber attached to each centromere going in both directions. I'm making a mess of this. There we go. So it's going to look something like that. So each diagram is going to be half a mark, by the way. So at this point, if you have the right number of chromosomes, half a mark, interphase, metaphase, it looks like we've got 1.5 marks out of 5 so far. So pretty good. Okay, and then the daughter cells, I made a little mistake here. What I meant to do is have 2 daughter cells because, of course, we are going to divide. So let me draw that second one here. There we go. By the way, if you want, you don't have to draw them like that. You could always draw them like this instead if you want to draw them side to side. So not a big deal. Either way is fine. Okay, so the daughter cells are, I'm just going to change the pen color again. Where's the pen pointer options? There we go. Back to red. Daughter cells, those sister chromatids are going to separate. So each cell is going to have 6 chromosomes. So there we go. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. You don't need to draw a nucleus or anything. If you, like I said, if you want to make them a little thicker like that to make it obvious they're chromosomes, that's fine. But I'm not needing anything fancy. In this case, I'm just looking that you have the right number, and that's it. I'm hoping that's easy peasy. Like I said, when we get to meiosis, it is going to get just a little bit more complex. And this is where people sometimes miscount and things like that. So let's talk about meiosis. Oh, before we talk about meiosis, I want to talk a little bit about regulation of the cell cycle and a little bit about cancer. So you probably know that we have different cell types, and it turns out that our different cell types do different things. Some of them grow and do mitosis kind of continuously. So that's like our skin cells. It's relatively continuously. Some cells, they do it when they're damaged or stimulated by other things. So the liver is a good example of that. If you take a biopsy of a liver, it will heal itself. Or if you scratch your knee, the tissues will heal themselves, right? Some cells kind of rarely actually divide and do mitosis. And this is all part of being a complex multicellular organism. And if we don't do this right, what happens? Cancer, other bad things. So we need to very tightly regulate these things. So I'm not going to really talk about this in a lot of detail this semester. If you take Biology 201, you're going to talk about this in a lot more detail. And again, you can look at it in the textbook. Some parts of it is interesting, some parts aren't so interesting. It's kind of one of those things that almost need an entire full another lecture to really do it some justice. And otherwise, I'm just going to confuse people, so I'm not going to go there. But I do want to just kind of mention a couple of things about this, the regulation. So you see, here's our cell cycle, right? We've got our G1. Remember, that's the growth or gap 1. And a lot of cells, of those animations out of order, a lot of cells actually stop there and will enter something called a G0 or a G0 resting state. And there's actually a lot of cells that will stay there for a long time. And they're not going to go any further, because notice the next step is the DNA synthesis. So, you know, we're not going to make a whole bunch of extra DNA if we don't need to. That's energetically expensive. It's not worth it unless you're actually going to divide. So depending on the cell type, some of them, they're going to grow and they're waiting until they're a certain size. They want to make sure there's nutrients available, that nothing is damaged, those kind of things. Most cells, though, are looking for this, these things called growth factors. So growth factor is kind of like an external messenger. So growth factor could be a hormone. Maybe you are a growing human, you know, we all start off small and we have these little growth factors that tell us it's time to grow, time to divide these tissues. And then there's general growth factors for different tissues. Some growth factors are stimulated when we get damaged or scratched or something like that. But often there's external messages that say it is time for this cell to grow. And then once it decides to grow, it goes past this checkpoint and it's going to divide its DNA. And this is the point of no return, because now it's going to exhaust a whole bunch of resources to replicate its DNA. So it's going to replicate its DNA. It enters G2. G2 is kind of another area where it's okay. Am I big enough? Is my DNA replicated? Is there any damage? And so on. And there's another checkpoint you can see there. And then once everything is good to go, it's going to start mitosis, go through the prophase, prometaphase, metaphase. Often it will stop just before anaphase. And like I said, there's some regulation that's checking to see everything is all the spindles are attached properly and all that. And then it continues. So all these have a whole bunch of mechanisms. And if they go wrong, it can be bad for the cell and bad for the organism. And so, like I said, one of the biggest issues with unregulated cell division is cancer. So just want to define cancer and talk about it for a couple of minutes, and then we'll talk about meiosis. Again, cancer is something I could spend a lot of time on, and unfortunately, we only have so much time this semester. So what is cancer? What is a tumor? A tumor is an abnormal growth. So if you take a look, there's that cartoon there. You've got normal cells, and they're growing, and they're all kind of uniform size and shape and whatnot, and then you've got this tumor here where the cells kind of look funny. A tumor doesn't necessarily have to be bad. You may know that most people, I think everybody probably has these benign tumors known as moles. Sometimes they're big and obvious, other times they're not. And a lot of people have at least tiny ones. And they're not really like, they might be an inconvenience if they're, in the wrong place, or maybe people think of them as beauty marks. It doesn't really matter. Benign means not harmful. There are also some that can be cancerous, and cancerous means they're causing some sort of damage to our body and causing this illness. This is just some pictures of some moles. Apparently there's apps you can get that can take pictures of your moles and decide whether they're cancerous or benign. Generally, you can see the benign ones are generally a little bit more uniform in size, like they're round, whereas the cancerous ones tend to be a lot more lumpy. So if you're concerned about that, get it checked out, by the way. If you have any moles that change color or size, that's another indication you should get them checked out as well. So let's talk a little bit about cancer. Cancer is, of course, a disease. So like I said, you've got a tumor, and the tumor is causing some sort of bodily harm, right? And that's where a tumor becomes cancerous, where it's actually causing you issues. So these images here actually represent the three most common types of cancer in Canada: lung cancer, breast cancer, and prostate cancer. So I thought it'd be worth it to show you those, and of course, they can lead to disease and death, and it's something that all of us. The statistics are that you have a 46% chance of getting cancer in your lifetime. So almost half of us are going to have cancer in our lifetime, and about a quarter of us are going to die from cancer in our lifetime. Those are the statistics for Canadians right now. So it's something that affects all of us. So here's just a picture. You may have heard of some of these terms, right? A tumor, it starts off benign, harmless. Eventually it becomes malignant, and that's where we start to call it cancer. And sometimes it's localized. And in many cases, the big concern is that cancer is going to spread. And the word for that is metastatic. And you can see it's in this image, it's going into the blood vessels, and it can spread to other body parts. And if it spreads to vital organs, your brain or your heart or something, that's where, and that's the huge danger around cancer. So if you can get it early, we can get rid of it, hopefully, and treat it. And the good news is, we've advanced really far. People are surviving a lot longer from cancers, and a lot more cancers are treatable than they ever used to be. So what causes cancer? That's the other big question. It turns out there's kind of two big things. One, which actually is not shown on here, but it is shown on here. The two big things are genetics, and the other big thing is your environment. So genetics, we can't control that. Some people have inherited, they're more likely to get certain types of cancers just due to their genes. And then everything else in environmental or lifestyle kind of things. You can see this is showing some of the cancer risks. The biggest one is tobacco. And well, actually, this chart is kind of misleading diet because it kind of depends on what it means by diet. I'll show you another chart here in a moment. But there's some big ones there, right? We've got things like radiation, tobacco, diet, certain types of viruses, and other things. And the huge long list of things that we call carcinogens. Here's a different chart. You can see tobacco use is the biggest one here. And they're saying that excess weight and obesity is number 2. So, you know, hopefully a little bit of encouragement for everyone to look after yourself. Lots of other things on there. You can see family history, alcohol use, those kind of things. So I'm not going to go into all these in a lot of detail, but probably some of these things you knew a little bit about already. So a quick question for you. What is a carcinogen? So you're probably thinking chemicals, but a carcinogen really is anything that is directly involved in causing cancer. So it includes chemicals, includes radiation, and some viruses. The other part relating back to Biology 107 is how do carcinogens cause cancer? And so the answer is that they directly or indirectly damage or affect DNA. And specifically, we're talking about DNA and genes that have something to do with the cell cycle. So if you mess up genes that are controlling mitosis, then those cells can grow out of control and cause cancer. So that's kind of cancer in a nutshell. And if you take more biology courses, you might learn a lot more about it. Like I said, there's only so much time in one semester, and we do have to move on. But it's a very interesting topic. I can honestly say it's something that I've learned a lot about, and every time I go away from it and come back, there's always new findings, and very fascinating area. So let's talk about meiosis. I'm thinking I have the right amount of time here. So just some definitions before we get to meiosis. First is asexual reproduction. So what does that mean? It means that 2 cells divide and they have the same genetic material in the end. So the term for that is clone. And so you can see, for example, there's some paramecium, and they're dividing by mitosis, and that's asexual reproduction. So second definition is sex, which is exchanging DNA. And we talked about this in terms of bacteria, and they were exchanging DNA using the sex pilus, and we call that conjugation. And it turns out Paramecium do this too. It's kind of interesting. You get some chance, read this little blog. It talks about Paramecium conjugation. One thing that we talked about in lab 5 is that Paramecium have 2 nuclei. They have a macronuclei and they have a micronuclei. And the micronuclei is a smaller nucleus, and during this conjugation event, they will exchange their micronuclei. And there's no reproduction involved here. It's just purely genetic exchange, and it gives them better genetic diversity, I guess. But check out that blog. It's kind of interesting. And then the last definition, of course, is sexual reproduction. So this involves the exchange of DNA, and usually we're talking about gametic cells that are joining, and those cells were created by meiosis. So we are animals, and you're probably familiar, or at least a little bit, with kind of how this works. So we have gametes, and the gametes, so that includes our egg cells, and our sperm cells are made through meiosis, and these things are haploid cells. And then at some point, if you reproduce, an egg and a sperm get together, this is called a fertilization event. So we have a haploid cell and a haploid cell, and they make a fertilized diploid cell. And that diploid cell becomes you, and it divides and divides and divides again a whole bunch of times, and you end up with a diploid organism. So this is the animal model. I'm not going to talk about all the other models. If you take Biology 108, you're going to talk about what happens in plants and fungi, and there's lots of weird things that go on. And even in animals, there are weird things that go on, because we can have haploid organisms. If you're a bumblebee and a worker bee, you're a haploid organism. And there's lots of weird things that go on, but we're just kind of mostly going to focus on humans and animals here. So what we want to do is talk about this event here, meiosis, and what is going on at a molecular level. So let's talk about that a little bit, but let's go back to these chromosomes, and I just want to review some terminology. Okay, so remember here we have a chromosome, and a chromosome is one unit plus its associated proteins, and it gets replicated, and we end up with two sister chromatids, and these are connected over here at the centromere region. So at this point, usually what I do, I have a box I bring to class, and people wonder what's in the box, and I pull out some candy, and I have some Mentos. And so I say, what is this? And people say it's a Mentos, and I say, we're gonna pretend it's a Mentos chromosome. So this chromosome is going to replicate, and there's two copies, and they're gonna be joined together, and I have an elastic band I put around, and this is at the centromere region. So I'm gonna come back to this candy analogy, and hopefully you find it useful. So I bring out the Mentos, and then I say, let's talk a little bit about what's going on in a human cell, a diploid organism. So here's a human cell here, and this cell has 3 pairs, so 6 chromosomes, 2 n equals 6. And so it looks like we got 3 chromosomes from mom and 3 chromosomes from dad. And so if you were human, you're going to have 46, 23 chromosomes from mom and 23 chromosomes from dad, but we're not going to draw that because that's too much. So it's easier to think in terms of 6. So each of these chromosomes is duplicated, and they each have they're a pair of sister chromatids, and they're joined at the centromere. So the last one to note is this term here, homologous chromosomes. So we want to make sure we get this straight and we're not confusing it with sister chromatids. So if you take a look, this is one of the chromosomes, and we've got 1 copy from dad and 1 copy from mom. So they are the same chromosome, but they're probably different, right? So they have, let's say this has the genes for, let's say, eye color. Maybe you got the blue eye color from dad and the brown eye color from mom. So it's the same genes, but different alleles. And so they're not the same chromosome, but they're homologous chromosomes, meaning they're kind of similar. So back to my candy analogy here in a moment. So at this point, then I go back into my box, and people get excited because there's even more candy. And I say, what is this? And it's like, it's Mentos, but it's not the same Mentos. It's homologous Mentos, or homologous Mentos chromosomes. And then this is not the end. I have more candy in my box, and I pull out one of these. And it took me a long time to find these mini Mentos, but I was so excited when I found them. And you get these little mini Mentos, and you can say, this is a Mentos, but it's not the same, it's not homologous. This is actually a different Mentos chromosome. Of course, there's a homologous mini Mentos chromosome. So anyway, this is my analogy I usually show in class, and as we go through and talk about meiosis, I flash the Mentos around. So let's talk about meiosis here and what's going on. So keep this in mind, this terminology. Like I said, sister chromatids versus homologous chromosomes. So first, just using the Mentos analogy, which stage of mitosis is this? And I'm hoping that everyone is thinking this is metaphase. And if you said that, you are correct. Notice there are no spindle fibers there, but you can imagine them in there. So let's talk about meiosis. Meiosis starts off just like mitosis. There's an interphase first, and it's the same interphase. So you have 2 chromosomes. You can see they get replicated, and now we have these sister chromatids. So exact same stuff, what we talked about before. And then we have actually 2 divisions. The first division we call meiosis 1. So in meiosis 1, the homologous chromosomes separate, but the sister chromatids stay together. So notice we still have 2 sister chromatids here, right? The homologous chromosomes separate. So 2 separations in meiosis. Meiosis 2, the sister chromatids separate, and we end up with less DNA in the end. So notice here, the initial cell, we have 2 chromosomes, right? So in this case, it would be 2n equals 2. That's the diploid cell. Over here, these are haploid cells. Okay, and n is still equal to 1, but they're haploid, so in the end we have 1. So we go from 2 to 1. We're dividing all of our DNA in half. So like I said, we'll do a sample here in a moment, and we'll see what I'm talking about in terms of just making sure that everyone can count appropriately for the mitosis meiosis question. So here we are, interphase. I think this is the exact same picture I showed you before. We've got the DNA has replicated, it's past the S phase. We have a couple of centrosomes, and we're going to move into meiosis. So meiosis one has its own little prophase one, metaphase one, anaphase one, telophase one. And then meiosis two is the same, except for they all have two after them. So here's prophase one. Some of the same stuff is going on. We got the chromosomes, they've condensed. We're kind of blend, for some reason we don't talk about pro-metaphase one. So you can see the nuclear membrane is dissolving. And there's one other additional thing that's actually going on here. So prophase one, they condense, and the homologous chromosomes actually pair up together. So you can see we have some homologous chromosomes. They found each other. They're like, hey, buddy, come over here. And then, just to try to give us a little bit more genetic diversity, this thing here happens. It's called crossing over. There are different terms for it, but our textbook is using crossing over, so I'm going with that. And so I just want to talk about that for a moment. I think, hold on, maybe I don't have that. Maybe there's a few more slides later. So crossing over, little bits of DNA are exchanged just to give us some genetic diversity. So I'll come back to that in a minute. Then we have metaphase one. Metaphase, things line up, but they line up so that the homologous chromosomes are still attached together. They're paired up together. So anaphase one is a separation, but we're not separating sister chromatids. We're separating homologous chromosomes. So you can see over here, we've got this one is going up and this one is going down. The sister chromatids stay together. So it's just separating homologous chromosomes. And then we have telophase one, and sometimes a nuclear membrane is formed, sometimes it's not. Sometimes we have cytokinesis now, sometimes it takes a little bit later. Depends on the cell type. In humans, usually it happens. So metaphase two, sometimes the chromatin gets kind of loose, sometimes it needs to repack, but for the most part, not a lot is happening during prophase two, other than if there's a membrane, it's going to dissolve, and more spindle fibers are going to form. Metaphase two is very similar to mitosis in that sister chromatids are going to separate. You can see everything is lined up. They separate, and we end up with our new daughter cells. And of course, since we've had two separations, divide into two, and then divide those into two, we're going to end up with 4 cells in the very end, and they are going to be genetically distinct. So I wanted to do a problem here in a minute and kind of show you what is expected for the test. But here's another test yourself. It says, what stages of meiosis are these? So let's take a look at the one on the left. So remember what these are. These here are homologous chromosomes. They're still together and they're lining up at the middle. So this here is actually metaphase one. Not sure why we like to use Roman numerals sometimes for these things, but it's a Roman numeral. So let's look at the one on the right. Notice I made my spindle fibers out of licorice. Thought you might find that cute. So in this case here, we have sister chromatids separated. So this here is anaphase two. So take a little bit of time to go over those. It's going to be helpful to know what's going on during the different stages. Like I said, the mitosis meiosis question, you're going to draw some of the things that are going on in these stages. So I'm going to come back to this. I want to show you the problem that I have for you and make sure that we have a chance to go through that. I may have to talk about a couple of these things 5 minutes into our review session next week. But what I want to do is show you this problem that and get you an idea of what you're looking for on the midterm. So here's the problem. This is the same problem I showed you before, right? This is n equals 3. So 2n equals 6, so 6 chromosomes. And we're going to draw these stages here, okay? So I'm going to change to the whiteboard, and we're going to draw these out. So where's the whiteboard? I had it here. Is that it? No, that's not the whiteboard I was looking for. Hold on a second. If for some reason there's two whiteboards in my system. Oh, Zoom, you're always, oh, there we go. Okay, so there's the problem. I cut and paste it. And I'm going to really quickly do my mitosis because we've already done mitosis. So we'll do it right here. Mitosis. And we have some metaphase, anaphase, and then what was the last one? Daughter cells. Okay, so I'm just going to quickly go over that one more time. And there we go. Okay, so the chromosomes I'm doing in red. So here we go. 1, 2, 3, 4, 5, 6. They're all lined up in the middle. And the spindle fibers all doing green again. There we go. So notice this time I just drew it sideways instead of up and down. Doesn't really matter. What matters is that you have the right number of chromosomes. So anaphase things are separating. We didn't draw that one before, I don't think, did we? So if you want, you can separate them like this. Or if you're trying to show a little bit of motion, you can draw them like these. And I'll show you how that's going to look in a second. 1, 2, 3, 4, 5, 6. 4, 5, 6. I barely had enough room to squeeze them in. So make sure you use a full piece of paper for this. I would recommend just to make sure you got everything squeezed in. So there's anaphase. So an extra stage there. We're showing things separating out there. And the daughter cells, 6 chromosomes. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So nothing new there. We basically had done that one before. Let's talk about meiosis though. And let us think about this a little bit. So meiosis, I'll do up here, and we're going to have Metaphase One. Will be the first one here. So Metaphase One, I'm going to draw this nice and big so that I can have everything there. Metaphase One, Anaphase One. Okay, we'll start there. So remember, we have the same number of chromosomes. Okay, the same number. So this is where people get messed up. Some people want to try doubling the DNA. Some people half it at this point. I'm not sure why. So remember, we have 6 chromosomes here. So there should be 6 over here. I just have to make sure I line them up properly this time. So this time the homologous chromosomes are together. So 1, 2, 3, 4, 5, and 6. So that's the difference there, is the homologous chromosomes are together. So make sure you got your terminology correct. There we go. And they're about to separate. So Anaphase One is just the continuation of that, where they're getting pulled apart. So I'm just going to make sure they're not touching anymore and draw basically the exact same thing. Okay, so Metaphase Two and Anaphase Two, and then the final cells. So we'll do that. So this is the big tip. Have the same number for both mitosis and meiosis, at least for starting out. And then when you get to the later stages, this is where other things happen. So we have Metaphase Two. So there's going to be one division. I don't know what happened there. Let me just erase that and fix that for you. Okay, for some reason, I can erase everything except for that weird triangle that showed up. So let me just cross it out, and I'll put Metaphase Two over here. Okay, there we go. So we have 2 cells, and we're going to have Anaphase Two, 2 cells. Oh, I like how it makes a nice circle for me. And then we have the daughter cells. That should be a D. So 1, 2, 3, 4. Okay, so back up here to Metaphase Two. So notice, I'll just draw a little dotted line. This is going to separate. Okay, this is about to undergo some cytokinesis. So we're going to have 3 in each of these, right? This is already at a point where we have 3 chromosomes. So we're going to, since it's metaphase, they're lined up along the middle, and they're going to look something like this. Drawing my spindle fibers. There we go. I must have some setting that's trying to fix things for me in this, and I don't know why it's messing things up for me. Drawing triangles and circles. So anyway, there we go. That looks a little better. Maybe not perfect, but we'll we'll have do. So notice they're lined up at the middle, just kind of like mitosis, except for now we only have 3 chromosomes. Anaphase 2, things are getting separated, so I'm just going to separate them out. Like I said, if you want to draw a little bit of motion, you can draw them like little V's like that, which shows they're being tugged over to the poles of the cell. Okay, those spindle fibers are in there. So remember, what's going to happen now is that these are going to eventually separate. And so we're going to have 3 chromosomes in each of the final daughter cells. And this is the good check, because notice over here we have 2n, that's going to be 6. Over here we have n that equals 3. So the haploid cells here, so these are diploid, which means they have double, and these ones are haploid, and they have half the amount of diploid. So there's my beautiful diagram. I hope you like that. So I know we are pretty much out of time here, so just give me 1 minute and I'll finish up. I know that I have a couple of slides here left on the PowerPoint, and I'll just have to finish those up next day. Mostly just a couple of definitions and a couple of things to compare between mitosis and meiosis, but it won't take too much of the review time. And then most of Monday, the rest of Monday will be a review time for the midterm. So good luck with your studying. Send me any questions if you have something you want me to cover on Monday in particular. And we're going to go through it all. It's going to be a bit of a whirlwind. And I'll have some office hours on the Tuesday before the midterm as well, if anyone has anything else to catch up on. Other than that, check out the review material I have on Moodle, and we'll see you next week.